Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami microbiologist. Today in this video lecture we are going to talk about osmotic fragility test of RBC, or we can say erythrocytes. Now my name is Muhammad Kamil Khan and you guys watching Kami microbiologist channel, so let's start the video. Now the main content in this video that we are going to talk, the first is we are going to see the introduction of osmotic fragility test. That what is actually osmotic fragility test. We are also going to see in introduction we are also going to see that what is actually the osmosis, solute, solvent, solution, each and everything. We are going to see hypertonic solution, hypotonic solution, isotonic solution. It is also important to know about that. Once we know about that, so then osmotic fragility test will be very easy for us. So we are going to see each and everything in the introduction part. Then we are going to see that what are the IM's purposes, importance or uses, means for what purpose, for why we use that test. Then we are going to see that who discovered this test, principle of the test, requirements, procedure, how to perform that test, normal range or what actually we get, means the result of the test, high and low osmotic fragility, means there are some condition and diseases in which the osmotic fragility is increasing and decreasing. So we are going to see all these, each and everything in detail. First of all, let's talk that what is actually osmosis. Osmosis is actually the process in which the molecules are moving from low to high concentration, and when the molecule is moving from low to high concentration through a semi-permeable membrane. So in this case the semi-permeable membrane playing a very important role, through which the molecule can be easily transferred from low to high concentration. This is osmosis. Then we have the fragility. So remember guys, fragility means the ability of something that can be easily broken down or destroy. Then we have RBC or erythrocytes. So all of you better know RBC or erythrocytes are actually the type of the blood cells that can carry oxygen to all part of the body. Then we have the test. So osmotic fragility test actually measure the RBC ability. In this test we mostly check the flexibility or the resistance of the RBC in different solution, mostly in hypotonic solution or in hypertonic solution. Means we just check that whether the RBC have the ability to stand in the hypertonic or hypotonic solution, means the RBC are rupture in these two solution or no. We are going to see these RBC in the RBC hemolysis in these two solution. Now let's explain the osmosis process through this diagram. As you can see, this is actually the solution. Okay, all of you better know that solution is actually made up of solute and solvent. Now these dot, as you can see, the dot is actually the solute, and solute is what actually the solute is the particles or something. Let's suppose in this case it is sugar or salt. Let's suppose it is salt that can easily be soluble into the solvent. Solvent is something that can dissolve the solute. So in this case the solvent is water. Okay, liquid or water, any part of the liquid, mostly water. So when we have solute and solvent, what we get the solution. So this is the solution, as you can see. Now in the mid, as you can see in this diagram, at the mid portion which is represented by the red dots, these are actually the membrane portion, and it is actually semi-permeable membrane. Semi-permeable membrane is actually the membrane which contain the pores, as you can see, and through this pores what happen, the molecules can easily be passes. So as you can see, this at the right side what we have high concentration. Why? Because in this case the particle amount are higher, while at the left side the particle concentration are low. So this is the lower concentration and this is the higher concentration. So what's going in this case, the molecules will be moving from higher to lower from lower to higher concentration through this membrane. So in the next picture, as you can see in the second beaker, what we see, as you can easily see that the water molecule can easily be passes okay through this semi-permeable membrane. So this is the low concentration, now it is passes from the higher concentration. This is actually called as osmosis. Okay, now this osmosis process was first of all discovered by Parpart et al. Et al means their colleagues in 1947. Then we have two other scientists named Vaughan and Suo in 2009, and they actually find out the osmotic fragility test on the basis of flow cytometric assessment. During this test, what actually they found the RBC measurement. They actually count the RBC measurement in the 0.9% of NaCl solution before and after the addition of the water, and they found that RBC are rupture or not rupture, means they just see the RBC maintenance in each solution, 0.9% of NaCl solution. Then we have the IM's important purposes or uses of the means for what purpose the doctor recommend this test. Mostly this test is specially used to just check the capacity of the RBC or the ability of the RBC, whether the RBC are stained in the hypotonic or hypertonic solution or not. And we are also going to see the morphology of the RBC through this test as well. This test is also used to find out the hereditary spherocytosis and thalassemia. Hereditary spherocytosis means when we have the RBC which are the spherical in shape. All of you better know the RBC are actually biconcave in shape. Okay, and in case of the thalassemia, what we have the body that do not have the ability to produce enough protein, or we can say hemoglobin are not produced, and these two conditions are mostly make the RBC fragile, means the RBC make it weak. Due to this way the RBC can easily be broken down. Now as you can see, this is the spherocytosis. Okay, now when the RBC are in a spherical shape, as you can see, this is the abnormal structure of the RBC. Due to this abnormal structure of the RBC, the water molecule can passes, and due to this way the molecule will not again release out from this cell, and due to this way this cell will be what happened in this case, the cell will be swell up and burst. But in normal case, as you can see, this is the normal structure of the RBC, which is the biconcave in shape. So in normal structure of the RBC, the water molecule can be easily passes from the cell outside and inside. That's why this RBC will not be rupture, although it rupture but not fast as the spherocytes RBC. Then we have the hypertonic, isotonic and hypotonic solution. As you can see, first we have hypertonic solution. So what is actually hypertonic solution? Remember guys, hypertonic solution is that solution in which the solute concentration is high but the solvent concentration is low, means the water concentration is low and the salt concentration, let's suppose, is high. We called it hypertonic solution. And when we put it the RBC, so what happened? The RBC will be shrink and destroy, as you can see. Now why? Because in this case the RBC is actually having the water molecule, or it is having the molecules, but their molecule is actually low they have actually low molecule, and in outer environment what we have high concentration. So if inside the RBC, if there is low concentration and in outer environment there is high concentration, so we better know that osmosis process will occur, and due to this way the molecule will be passes, means from the RBC the molecule will be passed to the outer environment. Why? Because the RBC have low environment, low concentration. So due to this way osmosis will occur, and due to this way RBC will shrink and destroy, as you can see. So in this case RBC will be shrink and destroy. Second, we have hypotonic solution, then we will explain isotonic. Now in hypotonic solution what we have less concentration of the solute but high concentration of the solvent, means water content is high but the salt concentration is low. We call it hypotonic solution. So in this case, what's the RBC have high amount of the concentration, means it have high amount of the solute, and in outer environment we have low amount of the solute. So what happen, the molecule will be passes from external environment to the internal environment of the RBC, and when the molecule is passes from the external, means from outside to the RBC, so what happen, the RBC will be swell up and the stage will come due to which the RBC will be burst. This is mostly occur in case of the hypotonic solution. So then we have isotonic solution. In this case we have the level is normal, because let's suppose at outer side there is a 4 let's suppose molecule of the water or we can say salt. Now then we have isotonic solution. Now in the isotonic solution what we have actually the water molecule. Outside the water molecule is present, as well as salt concentration is also present, but inside the RBC the same amount is also present, and due to this way what happened, the water molecule can be passes and it can be also released out, and due to this way the RBC can maintain their ability inside this solution, and the RBC will be there is no rupture in this solution. So this is actually the phenomena. Then we have the working principle. So remember guys, it work on three solution as we already talked. First we have hypotonic solution, in which the RBC are shrink, and we call that exo-osmosis. Okay, now as you can see, means this is exo-osmosis. This is called endo-osmosis, when the RBC take the water molecule, and due to this way it can swell up and burst. This is called endo-osmosis. When the water molecule is released out from the RBC and RBC are shrink, this is called exo-osmosis. Okay, so then we have hypertonic solution, in which the RBC in this case the RBC are swell up, okay, and in this case the RBC are shrink. In the second case RBC are mostly shrink. Now then we have isotonic solution, in which there is no changes in the RBC. So as you can see, these are the solution, and in each of the solution what we put the RBC. So the first solution is actually representing a hypotonic solution, and we added the RBC. So in this case what we have less number of the solute, as you can see, and due to this way the RBC is also having a large number of the solute. So what happened, the molecule will be moving from the low concentration to the high concentration, and due to this way the RBC will be swell up and burst. So it mostly occur in the hypotonic solution. So now then we have in the second picture, as you can see in the second picture, what we have isotonic solution, and this is the normal RBC, as you can see. So we have same number of the solute and solvent in the RBC as well as in the outer environment. So what happened, the molecule will be easily passes, and due to this way the RBC structure is maintained normal. Then we have the hypertonic solution, in which the concentration, as you can see, is high, but inside the RBC the concentration is low. So the means that the molecule will be moving from low to high. What happened, the cell will be shrink and destroy. Then we have the requirement for that test. I hope you better understand about the hypertonic, isotonic, hypotonic, each and everything. Now let's talk the test requirement. That what we required. We required test tube with rack. We required sample blood, but not in the ADTA tube due to the extra amount of the salt. Mostly we take the RBC red blood cell. Mostly we take the blood from in a simple tube, and then we have to keep it into the ice box to prevent it from the clotting. Then we need 1% initial solution and distilled water, or we can say normal saline. We need pipette or dropper. So these are the normal saline, droppers, and test tube with rakes. So these things we need. Let's talk about the procedure. First of all, we have to take 12 test tube into the rake, as you can see. Now in each of the test tube, what we do, we have to add water molecule. Three things we mostly add. First of all, we had the water molecule, mostly distilled water, and then we add NaCl solution, 1% NaCl solution, and we add blood. So three things we add. in all of the tube, what we add 1 drop of the blood. First of all, what we add in the first tube, we add 4 drop of the distal water. Then in the second tube, we add 9 drop of the distal water. In the third tube, we add 10, and then in the fourth, fifth, we are increasing the distal water concentration drop by drop, as you can see. Okay, and in the final 12th tube, we add 25 drop of the distal water. Similarly, if we see the initial solution, 1% initial solution, so we add 22 drop of initial 1% initial solution. So what we get the tonicity 0.88. This is mostly isotonic solution. Okay, when we add 4 drop of the distal water and 22 drop of the initial, so we get a isotonic solution. So when we are going from first tube to 12th tube, the distal water concentration is increasing, and when we are distal water concentration is increasing, while the salt concentration is means initial solution concentration is decreases. But when we are going from 12th to first tube, so what we see, we are seeing that distal water concentration is decreases, while the salt concentration or the initial concentration is increasing, and due to this way, what we get the tonicity. So this is how we adding the distal water and initial solution. Now once we add it, so then what we do, we have to shake it gently, and after that we put each of the test tube into the centrifuge, and we have to spin it, means centrifuge it at 3000 rpm for 30 minutes, for 20 or 30 minutes. After that, what we see, we have to check the result. Now in each of the tube, as you can see, in the 4th tube, as you can see, there is no hemolysis, means there is no rupture of the RBC. Why? Because in each of the tube, the tonicity is 0.5, as you can see. Okay, so it means that there is a salt concentration is actually, we have less high concentration of the salt, as you can see, in each of the 4 tube. But the distal water concentration is a little bit increased in each of the 4 tube. That's why the RBC are not ruptured, and we get clear supernatant, as well as we get the pellet. In the pellet, what we see the RBC. Similarly, if we see at this 6th tube, we see partially hemolysis, as well as we also see the pellet. Partial hemolysis means in this tube the color is actually pink, and in this means we can say that there is the RBC are starting rupture. This is the normal phenomenon. Mostly the RBC are rupture at 0.48 tonicity. If we have 13 drop of distal water and 12 drop of the sodium chloride solution, so RBC is starting rupture. So this is normal. Then we have in all of the solution, the RBC starting rupturing, and then at the last 3 tube, we have complete hemolysis, means uniform red color we get. So this is actually the color we mostly see. We can also check it by using spectrophotometer. Okay, now this is the second picture. As you can see, this is the normal result, as you can see. So at 0.48 tonicity, what we see the rupture of the RBC, which is actually the normal. But remember guys, if you see the rupture at 0.68, so it means that the patient have spherocytosis. And let's suppose if you see the rupture at 0.36, not at all the tube, you not found the rupture, or you not found the hemolysis, blood hemolysis, but at the 11th tube you see the hemolysis of the blood, it means that the patient is thalassemic. And if you see the hemolysis blood at this point, so it means that the patient is hereditary spherocytosis. A rupture of the hemolysis, how you will check hemolysis? Hemolysis, you will check it that the color is red. Okay, mostly the color is red, so it means that blood are hemolyzed. If the color is no red and you found the red color at the bottom, so it means the blood are not hemolyzed. Let's see the normal range. So hemolysis normal range is actually 0.4. At 0.4 tonicity, the blood are usually hemolyzed, so it is normal. Then If the hemolysis occur at 0.65, it means that it is spherocytosis, and if the hemolysis occur at 0.31 at tonicity, so it means that it is the thalassemic case. Then we have some condition in which the osmotic fragility tests are low and high. First we have the chronic liver disease, in which the osmotic fragility are low. We have iron deficiency anemia, thalassemia, hyponatremia, polycythemia vera, sickle cell anemia after splenectomy. Then we have condition in which the osmotic fragility test get. So now this is actually the osmotic fragility test, means the condition in which the osmotic fragility test or the osmotic fragility get low, and this is actually the condition in which osmotic fragility get high. This is the high and this is the low. Okay, not both low. Actually, this is high. Now, the condition in which the osmotic fragility get low, we have the hereditary spherocytosis and hypernatremia. Okay. So that's all about the osmotic fragility test. I hope you understand. If you like this video, so make sure to hit the like button, share this video with your friend. If you have any kind of question in your mind, you can write the question into comment. Inshallah, I will try my best to give you answer as soon as possible. Also subscribe the channel to get more interesting videos like that. Thank you so much.